welcome to the deep dive. Today, our mission is to step inside the just astonishing world of plant physiology.
Which is really just the science of how they work.
Exactly. We're moving way past the idea that they just, stand there and look green. We are going to try and decode one of life's greatest engineering feats.
I mean, they defy gravity, they manage this constant energy crisis, and they schedule their whole lives down to the minute, all without a brain.
It's incredible.
It is. And when you look at the source material, you see that plant life is this amazing integration of, well, physics and incredibly sophisticated molecular signaling.
So for this deep dive, we've broken it down into three big areas.
That's right. First, the foundational physics, how they move water and energy around. Then we'll get into the internal signals, the hormones that control their growth and shape.
And finally, the part that just blows my mind, how they measure time, how they know exactly when to flower.
Yeah, that's the really complex part.
And we really want to focus on the how today. How does a giant sequoia get water hundreds of feet in the air with no pump?
Or how do they solve that basic conflict of needing to breathe in carbon dioxide while at the same time basically hemorrhaging water?
Yes. And the big one.
Yeah.
How does a leaf, way down on a branch, send a message to a bud that tells it, okay, it's time to reproduce now?
It's a beautiful system. Let's break it down.
Okay, let's start with what you just said, that immediate crisis of just existing water management. The sources are pretty clear that the second a plant opens its pores, its stomata, it's in trouble.
It's an immediate trouble. It's a brutal trade-off that's just enforced by the laws of physics.
Because the pull for water to leave is so much stronger than the pull for CO2 to come in.
Vastly stronger. For a typical C3 plant, think your average backyard plant, the research gives this almost unbelievable statistic.
I know the one you're talking about.
They lose as many as 400 water molecules for every single CO2 molecule they manage to gain.
400 to 1. It's wildly inefficient. It makes their survival seem like a miracle.
It really does.
So, okay, given that insane evaporative demand, how on earth does a tall plant, a redwood, for example, lift water up against gravity? We're talking 300 feet sometimes.
And we know for sure it's not root pressure, right?
That's negligible.
Completely. It can't push water up more than a few feet. The whole system relies entirely on the amazing properties of water itself.
It's polarity.
It's polarity and the cohesion and adhesion that come from that. Water molecules just love to stick to each other, and they stick to the walls of the xylem. It creates this continuous column with incredible tensile strength.
So we're not pushing from the bottom, we're pulling from the top. That's the cohesion tension theory.
That is it, precisely. The tension is actually a significant negative pressure.
A vacuum, basically.
Yeah, think of it like that. It's generated right at the top, in the leaves, as water evaporates. It's like sipping water through a 300-foot tall straw. The evaporation creates the suction, and the strength of that water column allows the whole thing to be pulled upward.
And it's totally passive. Just physics.
Purely physical.
But for that to work, the column has to be unbroken. And before the water even gets into that xylem highway, it has to get through the root. So how does the plant control what actually gets in?
This is where the plant gets to be selective. As water moves through the root, it has a few different paths it can take.
The apoplast, the symplast.
Right. The apoplast is basically the space between cells. The symplast is the connected cytoplasm inside the cells. And then there's the transmembrane path, which is in and out of cells.
So where's the checkpoint? Where's the bouncer?
The bouncer is the endodermis, and specifically the Casparian strip. You can think of it as a waterproof gasket embedded in the cell wall.
So it blocks that easy pathway between the cells.
It completely blocks the apoplast pathway. It forces all the water and all the dissolved minerals to cross a living plasma membrane to get into the symplast, and that's the key.
Because then it has to be actively transported. The plant gets to choose.
Gets to choose what to accept and what to reject. It's a critical control point.
Okay, that makes sense for control. But what about just getting enough stuff? I mean, plants need huge amounts of nitrogen and phosphorus, and the soil isn't always rich in them.
No, absolutely not. And that's where symbiosis becomes so important. The sources point out that something like 80% of plants partner with mycorrhizal fungi.
The fungi basically extend the root system.
Massively. The fungal hyphae create this enormous web that reaches way beyond where the root itself can go. And they are fantastic at pulling in phosphorus and other metals in exchange for some sugar from the plant.
And then there's the really famous one, the nitrogen fixers.
Right, the rhizobia bacteria that form nodules on the roots of legumes. Nitrogen fixation is super energy intensive, and the enzyme that does it, nitrogenase, gets destroyed by oxygen.
Which is a bit of a problem.
A huge problem. So the plant has this brilliant solution. It produces like hemoglobin, which is an oxygen-binding protein, and it surrounds the enzyme with it. It keeps the oxygen levels low enough for the enzyme to work, but high enough for respiration to happen.
A perfect little microenvironment.
It's a masterpiece of management.
So the plant has water and nutrients. Now it needs to turn sunlight into energy. Our sources bring up the Emerson enhancement effect as the key discovery here.
That was the big breakthrough. Scientists saw that if you shine red light and far red light together, you get more photosynthesis than just adding the two separate effects.
Which means there must be two different systems working together.
Exactly. It led directly to discovering photosystem I and photosystem II working in a series.
And their job descriptions are pretty distinct.
Very. PSII is the water splitter. It rips water apart to get electrons, and it produces oxygen as waste. It's a really powerful oxidant.
And PSI?
PSI takes those electrons and uses them to make a powerful reductant, which is NADPH. And together, they generate the ATP and NADPH that power the whole show.
The chemical energy. But that brings us back to the plant's biggest vulnerability, right? The enzyme that actually fixes the carbon, rubisco.
The rubisco dilemma. It's the most abundant enzyme on Earth, and it's flawed.
It's slow, and it makes mistakes.
It makes a huge mistake. It's supposed to grab CO2, which is carboxylation, but it also grabs oxygen, which is oxygenation.
And that starts a process called photorespiration.
Which is just a massive waste of energy for the plant. It undoes a lot of the good work. And critically, as the temperature goes up, rubisco gets even more likely to make that mistake and grab oxygen.
So in hot, dry climates, this is a disaster.
A total disaster, which is what drove the evolution of these incredible carbon-concentrating mechanisms. C4 and COM.
Right. So C3 is the standard, inefficient model with that 400-to-1 water loss. C4 plants figured out a workaround.
They did. They used spatial separation. They fixed CO2 in their outer cells with a different enzyme, pepcase, that doesn't make that oxygen mistake. Then they pump that fixed carbon into deeper cells, the bundle sheath cells.
Where rubisco's waiting.
Exactly. And they flood those inner cells with such a high concentration of CO2 that rubisco has no choice but to grab the right molecule. It almost completely suppresses photorespiration.
And the water savings are huge. Down to about 150-to-1.
A massive improvement. But then you have the real extremists, the desert plants.
The cam plants, like cacti.
Cam plants. They use temporal separation. It's ingenious. They only open their stomata at night when it's cool and humid. They take in all their CO2 then, store it as an acid.
And then they shut down completely during the day.
Completely sealed. And during the day, they release that stored CO2 internally to rubisco. Their water loss ratio is phenomenal, sometimes as low as 50-to-1.
It's like they built 3 different engines for 3 different environments. But what about when a plant gets too much sun? That can be damaging, can it?
Oh, absolutely. Too much light is a huge stressor. The plant has to have a way to get rid of that excess energy safely. It's like a pressure release valve.
And that's the xanthophyll cycle.
That's the key. It's this rapid interconversion between a few different pigments. And when the light is too intense, the plant produces more of one called zexanthin, which is brilliant at just dissipating that extra energy harmlessly as heat.
Okay, let's shift gears now from energy and water to the internal command system. Hormones. We have these 3 concepts: growth, differentiation, and development.
Right. Growth is just getting bigger. Differentiation is cells getting a specific job. And development is the sum of both.
But the really mind-bending concept here has to be tutipitancy.
Oh, tutipitancy is amazing. It's uniquely plant-like. It just means that most plant cells, even after they're specialized, keep their entire genetic blueprint. They can, if you give them the right signals, revert back to an embryonic state and grow into a whole new plant.
Which is just not something animal cells can do.
Not at all.
Wow.
And those right signals are the hormones, starting with the first one ever discovered, auxin.
Auxin is all about shape and polarity. So how does it move through the plant in such a directed way?
It moves through what's called polar transport. It's strictly cell-to-cell, and it's unidirectional, mostly flowing downwards from the shoot tip. It's an active process using specific carrier proteins.
And its most famous effect is making cells get longer, the acid growth hypothesis.
Right. It's a really cool mechanism. Auxin basically tells the cell to turn on its proton pumps. These pumps push protons into the cell wall, making it more acidic.
And that loosens the wall.
It loosens the bonds holding the wall together just enough so that the internal water pressure, the turgor pressure, can stretch the cell and make it longer.
A simple but powerful idea.
Yeah.
And auxin's effects are everywhere. Apical dominance is the classic textbook example.
It is. The tip of the shoot makes auxin, it flows down, and that inhibits the side buds from growing. You snip off the tip, the auxin stops, and the plant gets bushy.
Simple enough.
But its effect on roots is what's really fascinating. It's all about concentration. A tiny, tiny amount of auxin actually helps the main root grow longer.
But too much is bad.
A slightly higher concentration, still very low, completely inhibits the main root, but strongly stimulates the growth of new side roots. So the same molecule can be a go signal, a stop signal, or a change direction signal.
Incredible. And then you have the cytokinins, which are sort of the opposite of auxin.
The yin to auxin's yang, they are all about cell division. The ratio between auxin and cytokinin is what tells a clump of undifferentiated cells what to become.
High cytokinin gives you shoots.
And high auxin gives you roots. It's the basis for all plant tissue culture. They also have that famous anti-aging effect.
Right, delaying senescence.
You can drip it on a leaf, and it'll stay green for much longer.
Okay, so last up for hormones, we have the stress and maturation signals, ABA and ethylene.
Abscisic acid, or ABA, is the big stress hormone. It's the plant's panic button. When the roots sense the soil is drying out, they send ABA up to the leaves.
As an early warning.
An early warning. And ABA is the signal that powerfully slams the stomata shut to conserve water. It also keeps seeds dormant so they don't germinate at the wrong time.
And then the simple one, ethylene gas.
The ripening hormone. It's why one bad apple really does spoil the bunch. We use it commercially to ripen bananas and tomatoes that were picked green. It also triggers what's called the triple response in a seedling that's growing in the dark and hits a rock.
It starts going up, gets thicker, and hooks around it.
Exactly, a little emergency maneuver.
So that brings us to the last, and maybe the most complex system of all, how the plant keeps its seasonal schedule.
Photoperiodism, measuring day length and vernalization, the need for cold.
And both of these very different inputs seem to converge on a single universal go signal.
They do. It's a mobile protein called florigen. In the model plant, it's encoded by the FT gene. In rice, it's HD3A. And here's the key. It's made in the leaves in response to the day length.
And then that protein physically travels through the phloem all the way to the tip of the shoot to tell it to start making a flower.
A message sent over a huge distance.
So let's look at a long day plant like a Arabidopsis. How does it know the days are getting longer?
It uses something called the external coincidence model. It's all about timing. The plant has an internal circadian clock, and that clock causes the gene constans, or Ho, to be expressed in a rhythm, peaking in the late afternoon and into the night.
Okay, so the clock is making the message.
Right. And in the long days of summer, that peak of CO expression coincides with sunlight. Light is the other signal. When light is present at the same time as the CO transcript, it stabilizes the CO protein.
And the stable protein is the trigger.
That stable protein turns on the FT gene at high levels, making a ton of florigen, and the plant flowers quickly.
The clock and the light have to happen at the same time.
That's the coincidence.
But short day plants, like rice, have to do the exact opposite. How is that system reversed?
This is where it gets so elegant. Rice has a version of CO called HD1, but its job flips depending on the light.
Flips.
Completely flips. In long days, when the HD1 peak coincides with light, HD1 acts as a repressor. It actively shuts down the florigen gene to prevent flowering.
Wow, so it's an inhibitor when the days are long?
Precisely. But in short days, when the HD1 peak falls in the long darkness, it suddenly becomes a powerful activator of the florigen gene. The same protein becomes an on switch.
And that switch is controlled just by whether light is present or not at the right time.
Just by the timing of the light, it's an incredible piece of molecular logic.
That is beautiful. Okay, finally, how does the cold requirement vernalization fit in with all this? For winter wheat, say.
Vernalization is basically a memory system. Winter wheat needs a long period of cold to be able to flower in the spring. What the cold actually does is turn off a major flowering inhibitor, a gene called VRN2.
So winter is spent removing the brakes.
You got it. The cold slowly removes this big don't flower signal. Then, when spring arrives and the days get longer, the photoperiod pathway we just talked about starts trying to turn on florigen.
And this time, it works.
This time it works because the VRN2 brake has been released by the cold. It ensures the plant doesn't waste its energy trying to flower in the middle of a false warm spell in winter.
The whole system is it's a series of checks and
Yeah.
balances.
When you look across all of it, what really stands out is this stunning integration. You have raw physics, like holding that water column together against impossible tension, and then you have these unbelievably precise molecular timers working together. A plant's whole life is just a masterclass in optimization.
It really is. We've seen how they use chemical stability, choosing a non-reducing sugar for long-distance transport, and physical structure, like how the angle of cellulose fibers determines which way a cell can grow, and then these light-gated molecular switches like CO and HD1. Everything is calibrated for survival.
Which brings us to our final thought. The sources note that the optimal temperature for photosynthesis is genetically determined, but also shaped by the environment. It can acclimate.
Okay.
But given the incredible precision we've just discussed, the fact that Rubisco's efficiency is so temperature dependent, the fact that the flowering switches in CO and HD1 rely on a perfect match between the clock, the light, and the temperature.
The whole system is balanced on a nice edge. It relies on temperature and light signals being in sync the way they have been for millennia.
Precisely. So what happens when global temperatures shift and that ancient, fine-tuned balance gets disrupted? The challenge for us is just monumental. How do you breed crops that can still read the seasons correctly when the environmental cues they evolved to depend on are constantly moving out of sync?
The plant's greatest strength, its precision, suddenly becomes its greatest vulnerability.